In this video, we will discuss about the dose-dependent study results of the best TEA-LNPs formulation. TEA is this T-cell mRNA efficiency and activations LNPs. LNP is lipid nanoparticles. So in the previous video, we have understood that they come up to the three different formulations that they named as TEA-13, TEA-37 and TEA-41, which shows the high transfection efficiencies. So they used these three formulations to check for the dose dependent studies whether the signals are because of these mRNAs that are inserted into the LNP or due to some other reason. So these things they have validated using the dose dependent studies. So in this case they also took the T cells, primary human T cells from the healthy donor, and did this dose dependent studies with their different formulations. And the mRNA that they use is the Firefly luciferase mRNA, and that is the reporter sci-fi EGFP. And the how this gives the luciferase signals is shown in this right hand side of the panel C. You can see that the mRNA will be exposed into the cytoplasm, and thereafter this Firefly luciferase protein will show this fluorescence signals. And so let us now come to those results of this dose dependent studies. So they did the dose dependent studies at three different time points, at 24 hours, 48 hours and 72 hours, and in all the 3 hours they are able to see a dose dependent increase for all the three formulations. However, the signal intensities for the formulation of TEA-41 is comparatively lower to the TEA-13 and TEA-37. However, they continue with their studies and also check the cell viabilities. And for the cell viability studies, for the positive control they use the PBS, and for the negative control they use the DMSO, as DMSO is known to affect the viabilities. And for all the three formulations at different dose dependent, they are not able to see a significant decrease in the cell viabilities, and it is comparable to the positive control, that is PBS, for all the three different time points, indicating that the high dosage of this TEA-LNP does not affect the T cells viabilities, and at the same time there is an increase in the fluorescence signals, indicating that their transfection efficiencies are dependent on the TEA amount or the TEA concentrations in the TEA-LNP concentrations in the reaction. So this is what they have finally concluded, that TEA-13 and TEA-41 seems to be performing better than the TEA-13 and TEA-37 seems to be performing better than the TEA-41. So this is, and these studies they are done on not just on the one healthy donor but on the multiple healthy donors T cells, indicating their reproducibilities, as we can see that error bars. However, in some cases the error bars are high, such as TEA-37 200 nanogram, but the results are still statistically significant compared to the controls. And so this is what they have shown in the main figure. In the supplemental figures they have shown some more results where they have checked this the size that are formed using this TEA-13, TEA-37 and TEA-41 lipid nanoparticles are comparable around 200 nanometers. If you want to check those results, you can go through the supplemental figures, although these results we have discussed in our previous videos. where they have also shown the nanoparticle size from this formulation and the other formulations. So that's all for this video. If you need to understand more about this dose dependent studies results in detail and wants to have a look into the supplemental figures, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.